Hello everyone, this is about the biotechnology principles and processes. See, biotechnology includes very important steps in our life also. Ever since the Rene Descartes, French philosopher, mathematician and also biologist of 17th century, all human knowledge, that is natural sciences, were directed to develop the technologies which add to the creature of the comforts of human lives as also value to the human life also. The whole approach to understanding natural phenomena became anthropocentric also, regarding the mankind at the central element of the existence also. Physics and chemistry gave rise to the engineering technologies and also industries which all worked for the human comfort and welfare. The major utility for the biological world is as a source of the food. Biotechnology, that is 20th century offshoot of modern biology, changed our daily life as its products brought qualitative improvement in the health and also food production. So further we continue. Herbert Boyer and also Stanley Norman Cohen combined their efforts in biotechnology to invent a method of cloning genetically engineered molecules in foreign cells also. By this discovery and its applications, Boyer and Cohen initiated what is now the multi-billion dollar biotechnology industry also. And in 1969, Boyer performed studies on couple of restriction enzymes of E. coli bacterium with especially useful properties. What they do actually, they'll be cutting the DNA of the two different strains and they'll be helping for us to understand the new DNA, how it is being formed. So Boyer observed that these enzymes have the capability of cutting the DNA strands in a particular fashion into the segments with cohesive or sticky ends in the strands. These clipped ends made fasting together pieces of this DNA a precise exercise also. Boyer's studies on the restriction enzymes would allow the Cohen to introduce specific DNA segments to plasmids and also utilize the plasmids as a vehicle for cloning precise, previously targeted strands of the DNA. Small ringlets of the DNA called plasmids, which float freely in the cytoplasm of certain bacterial cells and replicate independently from the cloning strand of the DNA, that is nucleide also we can call it. So Boyer and Cohen cloning methodology made possible to produce the synthetic insulin for those with the diabetes and a clot dissolving agent, that is streptokinase, for heart attack victims also, a growth hormone for understanding the or underdeveloped children also. So during this 1970s only, biotechnology emerged as a new discipline as a result of marriage of biological science with the technology. Here the meaning is biology included with the technology. So we usually call this as a biotechnology. It has been possible due to the revolutionary discoveries made in these two. Biotechnology is not a pure science but as an integrated effort of these two, the roots of which lies in biological science also. For the first time, Carl Erike, 1919, used the term biotechnology, a definition of biotechnology that encompasses both traditional values and also traditional views, and modern molecular biotechnology has been given by the European Federation Biotechnology, commonly EFB. Definition: biotechnology is the integration of natural science and organisms, cells, parts thereof, and molecular analogues for the products and services. So we have to use all these definition words also. If you see, the development of the recombinant DNA technology has allowed to modify microorganisms and other organisms to create in them highly valuable, novel, and also naturally non-existent capabilities. It is also called as genetic engineering. And Berg is often considered as the father of genetic engineering. He was awarded Nobel Prize in the year 1980. So further, if we continue in this session, we'll see some more information regarding the biotechnology is the science of utilizing the properties and uses of microorganisms, or they exploit the cells and also cells constituents at industrial level for generating the useful products essential to life and also human welfare. So in this sense, making of curd, bread, or wine which exploit naturally occurring organisms like lactobacillus and yeast respectively. And however, it is used in the restricted sense today. it to refer to such of those processes which we genetically modified organisms to achieve the same on a large scale also. So further, many other processes or techniques are also included under biotechnology. For example, in vitro fertilization leading to a test tube baby, synthesizing a gene and using it, developing a DNA vaccine, or correcting a defective gene therapy are all part of the biotechnology, which has taken one more step forward about the biology. In vitro fertilization technology was pioneered in the human professor Robert Winston also. The same technique was used by the P. Stepstow and also Edwards to produce the world's first test tube baby Louis J. Brown on July 25th, 1978. The European Federation Biotechnology has given a definition of that. That is, we have seen already in the previous matter. If you see, the techniques of genetic engineering include creation of the recombinant DNA, use of gene cloning, gene transfer. These techniques allow us to isolate and introduce only one or the set of desirable genes without introducing undesirable genes into the target organism. The first recombinant DNA from a plasmid autonomously replicating circular extrachromosomal DNA carrying antibiotic resistance gene in the bacterium Salmonella typhimurium. There are three basic steps in creating the genetically modified organisms or transgenic organisms: identification of DNA with desirable genes, introduction of the identified DNA into the host, maintenance of the introduced DNA in the host, and transfer of the DNA in its to its progeny. Thank you for watching. Please do subscribe my channel and share it if you like it. Thank you.